what is your favorite part of the cryo-ED workflow?
Yeah, I'm glad you asked that because I was thinking about that. I think for me, and I'm a biased vendor, let's be very clear about that also, but after a lamella is thin, bringing it back to fluorescence and seeing that your fluorescence still remains. There's so many places you could lose fluorescence along the way, so to keep it is an awesome feeling. Much more dim, obviously.
Okay, I think we're exactly on time, so here's Geoff from TFS. Please take it away.
So hi, yep, my name is Geoff Perumal. I'm one of the tomography specialists. I cover everything east of the Mississippi River. Been doing this for about 4 years with Thermo now, 3 years with Thermo now, 10 years in the industry. So I always tell everyone, please consider me a resource as well. Glad to pass out any information to stay in touch. But for me today, I'm going to talk a little bit more about the prep side of tomography. We'll really pick it up mostly after the freeze and everything that runs up to getting it inside of the TEM. We'll think about, what makes our samples ideal, how we go about creating them, and some of the advantages and some of the more recent releases of the ever-growing CryoFit portfolio. So for me, I always like to think of cryotomography not as an event that just happens at the last step in the TEM. And bear with me while I just pull up a laser pointer. But again, the entire workflow, because as the old bad adage goes, garbage in, garbage out. And so we want to make sure that every step of the way, we're ensuring that our samples are remaining vitrified, they're well taken care of, they're targeted if they need be. So when you get into the cryo-TEM, you feel the confidence that your ROI is in that lamella or your multiple ROIs are in them. So again, going from freeze through your plunge if it sells on grids, high pressure freezing if your samples are thicker or if you're going way of waffle, into the fib. Now, as has been pointed out twice today, and it's really wonderful, and not all tomography will require fib. It's one of those thoughts that are overlooked a lot of times. So whether it's off to the periphery of the cell, your sample is small enough, then we can all together go around it. In the instances where it gets thicker, and we'll define what thicker means, we will need some intermediate, and that would be a fib. So this is a very similar slide to what Alex showed before. So again, if you're paying attention. One of the really nice things about tomography is the flexibility in samples. Single particle, obviously you're working with isolated, really small proteins that are plopped down on grids. And that's basically the crux of it, right? Like, there's not a huge amount of variety in the size of your sample typically, though there is to a certain degree. But I mean, going from scales as small as virus up to tissues which are excised from animals is one of the nice things that we offer with cryo-electron tomography. Oh, excuse me. And as also was mentioned earlier, depending on the thinness is how we're going to treat them. So once we get to the point of, some bacteria through cells, particularly cells that are plated down onto grids, we're going to be creating windows, which I think everyone here knows is also referred to as lamella or lamellae. So you'll probably hear me say that a bunch. A lot of the research these days is going the way of waffle because of higher number of samples inside each lamella. Organoids have become increasingly interesting. So those two can also be sliced open with a traditional fib. Once you get to those thickest samples, and this is right at the end of Alex's talk when he really realized how much over time he was, we go move into tissues, and there's a bit of a different workflow for that one called cryo lift out. Got a couple examples of that here. But as was mentioned then, the idea of these multiple samples is that the process is more or less the same. Sample, cut it open, create a window, bring it into your TEM. Now, how you go about doing that is the parts that will vary. So samples, and this says up to 5 microns. The number could be 7. I would never say 10. Up to about 7 microns is usually my number for safeness in plunge freezing into liquid ethane. Anything beyond that one, we're going to really start to see some poor or just complete devitrified samples. In that case, then we'll slide over towards the high pressure freezer for your more efficient freezer of thicker samples. I used to do a ton of freezing of C. elegans in my previous life. My comment with it is, most people will tell you about 200 microns in thickness. I was always a believer, like never go to the edge of a technology for efficiency. So if your HPF is spec'd at 250, hit that like 175 range. It just helps ensure that a really long process is good from the first step. So I think I have to click this to get to everybody. So plunge freezing, again, the process, I think everyone here is familiar with it, either from single particle point of view or from traditional tomography point of view. Samples are brought down, are either polylysing down or adhere onto DEM grids. They're brought into a plunge freezer, which has your sample on it. We usually do a little bit of media-free rinse to kind of get rid of some of the excess gunk on there. And then the process of blotting comes in. So as was mentioned also earlier today, there's a bunch of different options for blotting. Ideally for tomography, you want to just back blot to kind of avoid everything getting run off. Once that blot happens, it's quickly injected into the liquid ethane. And now we have our presumably well vitrified sample that's noted here in this movie. Now, the next step is, again, dependent on what your research dictates. We're looking at a cell which is nice and flat, living on a grid. And I think I got my legend. Yeah, perfect. And if you place it directly perpendicular to that of a TEM beam inside with no tilts, this is about what you might expect to see. Off to the periphery where the cells flatten out, or certainly sub 200 nanometers, we have a lot of TEM penetrance. So you can see right through it, meaning that once we tilt, we're still going to be thin enough to get some pretty high angle tilting. But one of the big things which starts to really attract a lot of the cell biologists into this world is the tomography, which happens more towards the heart of your cell, the nucleus, where things get quite quick, quite fast. And that's exactly what we're depicting here in the middle of this slide, where that black is the lack of penetrance of your TEM, and this of your TEM beam. And this is where our Golgi is going to exist. A lot of those other fun, interesting organelles, which we're going to want to pass down through the tomography pipeline. So for that reason, we need to include that intermediate step. And that intermediate step is a cryofib. Now, there's a whole bunch of flavors of cryofib, as Alex also pointed out earlier today. Broadly speaking, there's gallium-based tools and there's plasma tools. A lot of more recent biology is going the way of plasma, but I mean, I've seen a lot of great work be done with the gallium tool, and in this case, it's our Aquilus tool, which is a standard cryofib. It does one job and one job really well, and it's cutting lamellae. So if you're working predominantly with just cells on grid, then this is the perfect tool for you. What happens is once our samples get a little bit more complicated. So waffle samples tend to be a little bit thicker than that of a traditional cell. The usage of plasma in any of these few species, xenon, argon, or some instances nitrogen, it shows a lot of benefit in a little bit more efficiently ablating your material. The paper which Alex mentioned earlier today also kind of implies that maybe there's a little bit less damage with plasma versus that of gallium. And then again, it's going to depend on which species you use your KB, but all in all, the plasma is showing to be a really nice benefit in this workflow. Now, I know you all just took your tour. I'm presuming you went down to the basement and you saw the newest introduction to the suite of tools here, and that would be the Arctis. I'm going to talk about the Arctis at the back end of it. At the back end of this, you can think of an Arctis as kind of like the combination of a cryo-TEM and a plasma fib. It's got the benefits of a smaller chamber, so we're going to keep contamination rates down. It uses the plasma sources, so similar to that of the PFib. It also carries an autoloader, so we're able to introduce 12 samples at a time into it, as opposed to the standard 2 grids, which most cryofibs can do quite routinely, meaning lots more lamellae. And with that, you would imagine a much larger dewar to allow us to run for 5, 6, 7 days. So I'm guessing most of you are probably familiar with what the anatomy of a fib is. I always like to kind of just go through it again. SEM imaging column is very similar to that of a TEM. You've got a source. Your beam runs through a high vacuum. It meets your sample at a coincident point to that of the focused ion beam. Now, at this point, your focused ion beam can be gallium or plasma. The angles are being more or less the same. Your distance from your pole piece will vary a little bit depending on the tools, but that coincident point will allow you to simultaneously ablate away your material and, if need be, image it with your E-column, which is a little less commonly done, but it's still going to happen. Software is the other part, which I'll just touch on really briefly. Sounds like there's a lot of experiments that are about to go into Tomo. The software for these tools certainly has reduced the learning curve. It's going to allow you to do a lot of multi-site lamella milling, as well as a GUI approach towards the actual the process going from cell down to thin down area. What we're looking at here in the middle is a yellow box, which is basically a mask above and below our cell, which we place our masks on our area of interest. And then from there, the software will basically mill away everything, turning our fried egg cell here into that end result lamella. So what does that look like if we were to go ahead and see them happen simultaneously? And I think I should turn off my laser pointer to play these movies. So up top, we're looking at that same image from the point of view of the fib, ablating away the cell. Now, usually there's a few different currents which are used. When you go from bulk down to 200 nanometers, you're able to get away with a lot of current initially to efficiently remove your material. But once we start to really thin down, as we're starting to see now, or we just saw, you want to reduce your current to make sure that you're polishing nicely. You're not leaving any streaks, which is known as curtaining in the lamella. The bottom video is simply the top-down view of the stem for both alignment as well as to let you see your cell going from full down to your 200, 250 nanometer lamella. Those lamellae inside the SEM are not going to be transparent enough for you to really make sure that an area which you might be targeting is there. We could certainly see that as we show here in A, and B, we could see the resultant, I guess these are yeast inside of the lamella here. So the quality of the lamellae, will allow you to do some really nice annotation, as we see here on the left, some segmentation on the right. But the advantage of this is going to be that we're taking our samples going from live to quick frozen, meaning that chemical fixation, dehydration, which you might do with room temperature samples, some of the artifact that produces is completely averted. The strength of filters will allow us to maintain some type of contrast in the lack of any heavy metals which go into these samples. So as I mentioned, the software is the other part, which is really important to allow us to go from what used to be a lamella or 2 lamella a day to tens of lamellae a day. Automation softwares is going to allow you to do that, and it's pretty much a standard on all of these microscopes. This set it and forget it idea. So whether you have a series of cells which are all identical, you could just plug your mask in over to numerous sites. The setup time will depend a little bit on how familiar you are with the software, but it's certainly the minority of this tool's operation time. It allows you to set it up, and long duration tours will allow you to let it run overnight and come in, presumably with a whole bunch of nice thin down lamellae. So this is what I sometimes refer to as the spray and pray method. So we're going to have a whole bunch of grids, our cells on a grid. We're just going to cut a lot of them. The next day, we're going to go and transfer it over to our TEM if it's available and scout each lamella. Well, does it have a particular area of interest? Did it stay vitrified? Did it stay put together? And it works. It certainly does. A lot of the tomography papers you'll see take this exact approach. But now as the idea of targeting becomes more and more of a necessity, there needs to be a better method for it. And there's been a couple of people here who presented it. And that's the idea of internal fluorescence. Internal fluorescence, I know he kind of glanced over it, but was actually designed by Alex DeMarco. I have a picture of his initial internal fluorescent microscope, but the idea of it is exactly as this sounds. We're placing an objective, excuse me here while I get my laser pointer again, directly inside of the chamber. It runs along the same line as your ion column, which you see back here, this silver post. Your SEM column is right here. So internal fluorescence on the aqueous, on the Hydra, happens inside the microscope, but it requires 180-degree rotation of your sample. So fine alignment is quite necessary for this. The idea of it, as you would imagine, is to scout out your cells, define an area. Now this is just mitotract sample here, so this green cluster here. But to go from bulk all the way down to your 180 degrees and confirm that that final lamella, as we see here, contains your area of interest. So again, that value is going to be that you're not just cutting 100 lamellae and hoping, before you get there. All the coordinates tend to transfer over from your fib into your TEM. Mapping softwares will assist you there, so you can go right back to that same lamella. Depending on what you're using, you can even know the tilt angle, so that when you do your back-end tomography, you have an idea of how much tilting you can get away with. So as I mentioned, IFLM is an ever-evolving product, starting with Alex's original tool, the PiScope. I was hoping he'd have eyes on his hydrant downstairs, but he doesn't, but maybe we could buy that for him. But from there, about a year later, the first productized IFLM was released in about 2019. More or less, it was identical to Alex's design. He helped with the implementation of it. And then from there, about a year and a half later, the current model IFLM, the one that's on the Oculos downstairs, was released. And there were a few more modifications. The objective was standardized at a 20x objective. The filter set was finalized for that one. So, and that's kind of where we live today. With this, now you're going to see a lot more automation happening on the software side of the IFLM today. But basically, there's two approaches that you can apply with IFLM. Now, the first one is the standard one, which you would think of, which is this manual approach of find, identify, and then shuttle back and forth between your IFLM and your FIB beam to ensure that your area of interest remains in there. That's something which I still think is applicable today, especially if you have a really rare fluorescing event. There are some automation, which I'll talk about in a little bit, but my tip for everyone, if you have a very rare fluorescent event, is to never go the automation way. And, it's a little manual, but watch your milling happen and check back as often as necessary. And the process would kind of look like using CHO cells, again, marked with mitotracker, is to identify, in this case, just a cluster of mitochondria, begin the process of a milling, at about your few microns chunk, bring it back to the IFLM. The return point is all manual, so you don't have to do much driving to go back to the IFLM. Confirm that your area still exists, bring it back, and thin down until we get to, in this case, about 250 nanometer lamella. At that point, again, as I mentioned before, the software will carry over a lot of your coordinates, the fluorescence data as well, so we could do some rudimentary alignment of our lamella in the TEM as well to go back to that area for your high-end tomography. Now, one other thing I'll point out here is just the overlay of it on once we bring it over, and then again to the ultimate TEM image where we can now really start to see the mitotract bead or the mitotract area until we get to the actual TEM itself. One thing I'll also point out is resolution. So obviously, even a fluorescent microscope is never going to have the resolution of that of a TEM. Even with these large cameras, we use a 20 megapixel camera. So we could actually use the pixelation imaging, as we're showing here on the bottom right, to your advantage to identify a fluorescing event in Z. Now, by that, what I mean is, again, a wide field solution will typically never have a resolution better than about probably 1600 nanometers in Z. So how do we take a system that has a 1500 nanometer resolution, fit it into a 200 nanometer lamella with some type of precision? And so what we do is we use a center of mass theory. So this pixelation is, like I said, going to be to our advantage. And so the IFLMs all do have a pretty good piezo drive on there, down to about 50 nanometer steps. So we're certainly oversampling our ROI. But what we could do is we could watch our pixels blink on and blink off. Somewhere in one of these, well, we usually use about 200 nanometer steps, we could identify your blinking pixel, which is associated with a slice number from your IFLM stack. That stack or that blink on that particular plane is then transferred over into your FIB for some automated milling. And how that looks, and I'm sorry if I'm butchering what I'm saying here, is essentially we identify an ROI. So in this case, somewhere around here, that particular slice number is transferred over to the eyes of the FIB. And basically what we're going to do is then there's some basic trigonometry, get you back to the exact area of interest in your cell. And I'm going to use the word exact a little loosely there. It's going to depend on how well you align it, how strong your fluorescent signal is to get you back to the precise position of it. But essentially what we're doing, and you might have seen it in a couple of images, and I think I have an example of it later, is we're milling an X into your grid bar, which is kind of known as height 0. Using that height using the height of your Z stack relative to that height 0, we know what plane it is, what plane it's in, and that trigonometry angle will basically tell the FIB exactly where to mill. All of this information is then passed over into that same software, AutoTem Cryo, which will allow you to then go ahead and begin a semi-automated approach This is a nice example here from Dr. Elizabeth Wright's lab at University of Wisconsin, where this exact experiment was done, in this case, using a couple of fluorescing yeast. So if you basically were to do just a simple SEM image and look down, you could certainly identify your cell of interest, but what's happening inside is where the IFLM comes into play. If you use just the IFLM, and I hope you can see it in this one, there's a couple of green dots right here. Those are fluorescing lipids. So this was the target which they wanted to aim against. So using that exact same approach of basically stacking down, finding the middle of mass of this, they were able to, in a semi-automated fashion, go from full cell down to 200 nanometers. The semi-automated part of it is a key part. And you add, similar to the manual approach, we will stop at about 5 microns, 4 microns, ensure that your fluorescence is still there. You can nudge up or down a little bit if there's any slip. And then from there, they went from a couple microns or a few microns down to 200 nanometers completely unassisted. That resulted in lamella. Then all of its data was passed over to the TEM, where we could see a nice alignment here. So all these suitable samples, we talked a lot about on grid. I'm going to just really briefly touch on lift out because I think all science kind of works that way to what's going on in the animal model at some point, which means tissues, high pressure freezing, and there is a solution for it, and that is the cryo lift out approach. So there are examples where cryo lift out has been done in cells on grid. It's not too common these days. It's more commonly done with high pressure frozen samples, such as C. elegans, as this cartoon is showing here. Idea is basically the same. We're going to go from bulk down to 200 nanometers. Where it differentiates itself is there's a needle, as we see here, it's the lift out needle. It's on the oculus. I believe it's on the Hydra as well. It's always in the same position. The needle will come right down to your specified area of interest once you identify it. And then there's 2 approaches. So as Alex mentioned, cryo lift out is a really dirty approach, or has traditionally been. That's because to get this needle to weld onto your chunk here, you usually are using one of your gas injector systems, and it's super messy. There's a few more modern approaches where you're actually sputtering some metal. I think I have it on the next slide. And you're basically creating intermediate to lift out, and then we're just using metal sputter rates, which are here really well. So you're getting around GIS, so you're speeding up that process. It's a lot cleaner too. The last thing I'll mention on this before I move to the next slide is these trenches here are really time consuming. So when you're doing traditional on grid, as you see here on the left, we don't really push the limits of these fibs to their highest currents. You get to a decent current to speed it up, but you're not really pushing it. When you create these lift out trenches, you really juice up your current, because just getting to this point here, and we haven't even lifted it out, it takes several tens of minutes with a traditional gallium 2O. This is one place that plasma is going to be really effective. The high current that you can get with plasma will turn what used to be tens of minutes into under 10 minutes. From there, and I think I have it in this next slide, let me just check, okay, cool. From here, after we lift out our chunk, we bring it onto a receiver, which is known as a half moon, and we can see that full image here. Note that at this point, this is not your 200 nanometer lamella. This is probably a micron and a half thick. You don't do your thinning for lift out until you've adhered it onto your spoke of your half moon. And it's basically done to ensure that a thicker piece of material can more easily mount onto the spokes, but also to, when we slice it away, we don't want any gunk getting back onto it. I think in this case, this is fairly true with most lamellae, but especially with lift outs, is get into your TEM immediately when you do it, because these things are the biggest cold traps in the world. The contaminant is just super attracted to it. So here's the same example that Alex was showing. This is that, this is the next level of lift out. It's this paper from Let's Go Group, I believe this, where they're doing serial lift out. Now, something to note about lift out is you go from a full worm down to an area, or whatever your species might be, down to a micron chunk, down to 200 nanometers. It's super wasteful of your material. Serial lift out is a way to more efficiently use your high pressure frozen material. Again, using a traditional TEM grid as your receiver, everything is welded down onto the spokes of your grid, and then you do some thinning. So there's still a loss to it. There absolutely is. You're going from 500 nanometers of micron down to 200 or so nanometers, but it's much lower loss. You can also, as you can see here, implement IFLN for some finding of your sample of interest here. Back into our tomograms are still going to be quite high resolution. So last kind of segue over is to where we are today and what we're going to be, taking a lot of tomography moving forward. And that's more towards full automation geared towards a TEM style fit. So if you saw some of the first cryofibs to really ever be released into the wild, they were truly just a traditional fib that had a cryo stage added onto it. Maybe they had a GIS, maybe they had a materials lift out needle. And so with the more modern tools, came the specialization in software. The Arctis, the half TEM, half cryofib, is really that next generation. So first thing is the integrated fluorescent microscope is now at a more optimal location. We did away with an SEM stage altogether, allowing you to place an objective directly below your sample. It's driven through a compu stage. As I mentioned, we were able to put an autoloader in there as well. So we're going to be doing about 12 grids at a time. With that would come obviously a longer duration. So these tools are expected to get 5 to 6 days. Still going to have a sputter target because you still have to code it. We use a PFib head on this, so for more efficient ablation. Again, potentially the idea of a little less dose damage on there. There still is an SEM column on here. The idea behind this tool is to really churn out lamellae. So you're not doing so much imaging or slow dwell speed imaging as rather alignments with this tool. But you could, I guess, in theory, do that as well. Cryobox on there also will keep the levels super low. So yeah, again, the idea behind this is not just high number of lamellae, but high quality, high yield lamellae. So in this case, we're looking at 15 sites, which were identified through the automated software. In this case, they basically just applied the same mask over to every single one. And then that basically walk away from the tool altogether. So the software will also, as you can kind of see here, build in your relief sites so that the lamellae are not overly stressed. And then the process of going all the way down to sub 200 nanometer routinely will be another nice benefit of this tool. Now here's an example of longer duration. In this case, 47 lamellae in about 48 hours, all done through the automated software. So you set it up. The other nice thing about this, by the way, is for those of you who've used any of the thermo cryofibs in general, you know that you do your fluorescence with the fluorescent software, you do the milling with AutoTem Cryo, you do XT for some importation, and then you do maps for your your combination of everything. This will all run under the same software platform, so there isn't transferring of data from software 1 to 2 and 2 to 3, which will also help in your efficiency. The last 2 bits on this one is, so we're offering a solution for a lot more lamellae. The next step, which should make sense, is to improve how the samples are loaded into the tool. I mentioned it does hold, it's a cassette that does hold 12 auto grids. With the Arctis will soon eventually come the Tomo grid. So again, for anyone here who's done some tomography and moved your grids, I'm sure all of you are used to putting a little Sharpie dot somewhere on your auto grid. Make sure that when you transfer it, you have some rudimentary alignment, but it's never perfect. It's usually good enough, but it's never perfect. And one of the main ideas behind this Tomo grid is to eliminate that step. So in lieu of a completely round auto grid, there are wings on each side of these. So that means they can only go in and out in essentially, well, 2 directions in theory. But it ensures that you're not, if you do ever take your grids out and reload them, that you're not going to have to find that little dot and hope that we're perfectly centered with it. This is going to open you up also to allow you to be 90 degrees from your milling angle, so you could really get those higher tilting angles when you bring it into the cryo TEM. Last little bit on it is, the Arctis, since it is this, next generation tool, a lot of the development has gone very quickly into this tool on the integrated fluorescent microscope side. So one thing I failed to mention before is that we actually use a different objective on the Arctis than that of the Hydra and the Oculus. Hydra and Oculus use a 20x objective with a 500 micron field of view. It has a 1.3 millimeter working distance And it's that last part, that working distance, which is why we use a different objective. To get the triple point, we actually need a little bit longer working distance. So this uses a 100x 0.75 4 mm working distance. 100x sounds great, it's a little bit more zoomed in. 0.75 is marginally better in resolution, but the cost is your field of view. The 20x has a 500 micron field of view, meaning that 4 images will get you that middle millimeter squared. With the 100x, the field of view is about 150 microns. So considerably smaller. And now the reason I point that out is, again, alluding back to the fact that a lot of the development has already gotten into the IFLM. Stitching is a feature that is standard on the ArcGIS. So we could do tile sets across the entire grid if you like. My comment on tiling is bin wisely. Binning at one is a super time consuming thing, and it's something that you should probably never do unless you get to your area of interest. Bin at a factor of 4, you can more efficiently identify your areas of interest. And then once you really identify them, then you could bin down to 1. And then that's where I would do my stacking. Automation on this is also going to be a little bit more improved than that of Hydra and Aqualos. There's not too much is different. The same approach is applied. The biggest difference is a second fiducial mark, which you see right here, which will allow our ion beam to be a little bit more precise than that of Hydra and Aqualos. Also, the way that the alignment is done is a little bit different with the ArcGIS. We're using the eyes of the fib more, and the eyes of the SCM less, meaning that we're more on plane, which is also going to only help improve the targeting accuracy. As we see here, we could nail a insulin granule, which is somewhere between 60 and 100 nanometers. So targeting should be better. And here's just a couple of tomograms from ArcGIS from a resultant lamella, which we can see here. So any of your favorite organelles. And I think with that, that is my last slide. I always did used to like to end these presentations with a nice zoom of the facility or a facility, but you all just saw that quite obviously. The idea of interconnectivity, as you can see here, take out your cassette from ArcGIS, bring it right over to your Krios. So minimize any of that transfer. There's no need to reload grids as well. So I think with that, I will stop. I'm glad to take any questions, comments, thoughts. I'm here for the next 2 days beyond today. So if anyone has any ideas, I'm more than happy to chat with you. Thank you all.
You got the EMO buttons. We actually disconnect them.
Which buttons?
The EMO ones, emergency shut off.
Oh.
So like, if you see our ArcGIS, there's a whole duty, don't have that button.
Oh, I did notice. That's why I looked different.
Yeah,
Okay.
there's no red note.
We disconnect them all.
Yeah.
Probably shouldn't tell me that.
We signed the waiver, so fine.
But any questions, general comments?
The tomo is commercially available yet?
Second half of this year, they're finally coming. And with that, there'll come a alignment on your Krios stuff. So whichever tool, because you have to account for the wings in there.
I will make the last side the same, so that only one side is shifted. So now you have the 5 angles of same grid,
I'm sorry?
the grid.
Let me say.
You have 2 positions that you could go in,
Yeah.
up or down. So if you had a third side, so it's easier. So this is the easiest one.
I'm sorry, I don't
So the
follow.
last angle, so there is only one position that you would go into the grid.
Yeah, so going back in one position is actually ideal,
Yeah.
because then when you get to the TEM, you know that you're 90 degrees from your milling angle. So, but again, that's why I said in theory, you could put it in 2 ways, because you can put it forward and backward. But I mean, the ideal situation is go right from the fib to the TEM, but if you did need to go back.
So these grid compatible with the regular current, or they can the winning have
With the shuttles.
turbines?
So the short answer is it could be. So depending on which cryofib you own, which is not an ArcGIS, some of the shuttles are back compatible to handle these tomo grids. Basically, you have to account for the slots. If not, let's say you have an Aqualos, you could buy the newer shuttles, which would have them in there. basically all the newest hydras should have them. So that should be accounted for. Arctis, of course. The older ones you should be able to retrofit onto.
We can fit in the Titan players.
I'm sorry?
I don't, they can fit in the play.
Yeah, the tomograph, absolutely, 100%.
So I have to do any modification.
Possibly. Depends which Krios you have.
How about G4?
It should fit, you know, again, you just need to do some alignments on there. There might be a modified cassette, which could come with it. If it does, it'd probably be, you know, free for any tools that it doesn't exactly fit with. And then you just have to realign your grippers to accept tomographs. And the common follow-up question we get to that one is, well, what about traditional auto grids? If you make the correction for tomo grids, you'll be applicable over towards traditional grids as well.
Thank you.
Thank you.